So as Dr. Santosh was mentioning that the series has already been started, and I was telling Santosh ji yesterday that ideally my talk should have been before his talk so that I could have give you some basic ideas about lipids and membranes so that follow the subsequent talks will be much easier, but that doesn't matter. We'll go back and forth, and please feel free to interrupt me at any point in time, and I'll leave my slides after the end of this series with Dr. Santosh so that you can have access to it, and I will also give you the references, and I would urge you to have those papers if you have some interest in membrane biology or membrane biophysics. All right, with those few words, let me get started with that. So as I was saying that I think Dr. Santosh has already started talking to you about organelle communication and membrane traffic. What I will be talking to you today is slightly take a step backward and talk about membrane organization and basically how lipids are organized and what kind of forces and thermodynamic factors are responsible for maintaining the organization. I'll be briefly talking about today, and also briefly touch upon what are the different modes of dynamics which keeps the cell membrane sort of active, because as Richard Feynman said, that everything in nature can be understood by the jiggling and wiggling of atoms. So membrane or lipid bilayer is no different from that. So in order to understand the physiology that takes place in membrane, we have to understand the dynamic aspects of the lipid bilayer membrane. So I'll try to introduce to you briefly on that aspect too. And then the following lectures will be more on what is called a lipid-protein interaction and membrane organization, membrane protein organization, sorry, and then membrane remodeling. So when I'll be talking about membrane remodeling, you find slight overlap with some of the things that Santosh and other will be talking about. So in that sense, we'll go back and forth. All right. So, the cell membrane is nothing but, I would say, the most indispensable part of a cellular life. Think of any form of cellular life, for example, bacteria, plant cell, or animal cell. You cannot think of them without thinking of membrane. Even Archaea or Archaebacteria has a membrane, and which has a very special kind of lipid. I'll also briefly touch upon that. The point is that the cellular life is indispensable without a membrane. And of course, the membrane is different in different organisms, be it in bacteria, gram-positive or gram-negative, plant cell, or an animal cell. Even some of the intracellular organelle has also are also membrane bound. So the basic organization of all these kind of membranes are the same, a lipid bilayer. However, the composition and their functions are different depending on where they are coming from. I just want to give you another example. Even the mighty virus, that we are all familiar with, and we are paying, because our life has been turned upside down because of this 100 nanometer object, that too also has a membrane. As a matter of fact, this membrane for SARS-CoV-2 and like other enveloped virus is acquired from the host cell. But the point is that you can now understand the importance of the cell membrane or lipid membrane, not only from the cellular aspect or cellular point of view, but also how an infectious agent like the SARS-CoV-2 actually can infect ourselves has a lot to do with its membrane. In fact, the spike glycoprotein against which the vaccines are raised is actually a membrane protein. So that's why membrane or cell membrane or plasma membrane plays such an important role in biology and cell biology, not only to understand the basic physiological process, but also to understand certain pathophysiology, for example, virus entry to host cell. So in order to design effective therapeutics and prophylactics, we need to understand the organization and dynamics of lipid bilayer membrane. All right. Now, I have already said enough why the membrane should be important, but it can still ask why bother, because this is only a 5 to 6 nanometer barrier, which basically separates the cells from the outside to inside world. So why should we be interested in studying biological membrane? Apart from the fact that I just told you that even the virus also carries a membrane, and we should learn that in order to design better antiviral and therapeutics. Other than that, there are certain central physiological processes that is dependent on the membrane organization and dynamics. Firstly, membrane or the lipid bilayer membrane or plasma membrane mostly constitutes a cellular boundary and establishes a connection between the extracellular and intracellular environment. In fact, it has been argued that formation of biological membrane or lipid bilayer is the first step in the origin of life problem. So it is argued mostly based on the work of Jack Szostak and Dave Dimmer that probably synthesis of lipid-like molecules was the first step in the evolution of life. So this is that important, right? So our life starts with membrane. Secondly, cellular membranes are important for maintaining cell-cell communication. Cellular entry, particularly nutrient uptakes, all happen through endocytosis process, and that involves membrane. Similarly, some of the cellular products, when they are released outside in the form of exocytosis, that's also a membrane phenomenon. For example, neurotransmitters released at the neuromuscular junction is a membrane phenomenon. So that's one of the role of the plasma membranes. And it has been estimated that roughly of all the biological processes that happen in a cell happens actually on membrane. I will slightly paraphrase it. It's not necessarily on membrane, but in and around membrane, and such that the membrane properties has a huge influence on those processes. So from that point of view also, plasma membrane plays a very central role. And lastly, and this is again re-emphasizing the point that I just made in case of viral membrane, that it is indispensable for functioning of membrane proteins. Now, why it is so? Because membrane proteins are actually embedded in membranes, so the physical properties of lipid are crucial for functioning of membrane proteins. This is not only important for normal physiological processes, but this is also important for the pathophysiological processes. Meaning, if you look at the druggable protein, that means the proteins that are targeted for drugs for any disease, a significant portion of that is actually membrane protein. So, and of that, a significant portion is actually what is called GPCR or G protein coupled receptor. These are nothing but 7 transmembrane domain membrane proteins. They are embedded in the membrane, so that means their functioning is actually influenced by the membrane physical properties. So, in order to design a drug, you have to also keep in mind that these are membrane protein and their functioning may not be as same as soluble proteins or secreted proteins. So from that point of view also, studying or understanding membrane physiology or membrane biology or membrane biophysics plays a very important role in contemporary cell biology. Now, coming to the thermodynamic factors that are responsible for the formation of lipid membrane or lipid bilayer is nothing new. We have already known in case of proteins that the hydrophobic effect plays a very important role. And I'm sure all of you are familiar with what is hydrophobic effect, and this is nothing but a manifestation of a very particular structure of water molecules in liquid water, which is a tetrahedral hydrogen bond network. Now, water has a unique structure because it has both electropositive hydrogen atom as well as electronegative hydrogen atom, and they can be engaged in hydrogen bond in the fashion that is shown here. Now, the biomolecules that we come across, those who are capable of engaging with this hydrogen bond network will be liked by water molecules, or they are called hydrophilic molecule, whereas those molecules which do not have the ability to interact with water molecule, or in other words do not have the ability to form hydrogen bond with water molecules, actually don't like water for thermodynamic reason, so they are called hydrophobic molecule. And this is again, as we all know, that whenever there is a bond formation, there will be a lowering of energy. So if two molecules are brought together, if they can somehow be engaged in bond formation, the whole system will be stabilized. Otherwise, there will be demixing, which is not a thermodynamically happy situation. So in that case, what will happen? The molecules which cannot interact with each other will try to separate from each other, right? And this is the basis of hydrophobic effect. So basically, since water molecule has a very particular structure, it is actually very clannish in nature, or very xenophobic in its nature, meaning it will not accept any molecule which do not have the ability to form hydrogen bond with it. So that means all the hydrophilic molecule has somehow have the ability to form hydrogen bond with water. All the hydrophobic molecules that are actually do not have the ability to form hydrogen bond with water. Now, a unique situation arises when we have molecules which has got both hydrophobic part and a hydrophilic part. That means some portion of the molecule are capable of interacting with water, some portions are not able to interact with water. Then what happens is a kind of a unique situation, and that is basically the driving force for all kinds of organization in biology that you come across, be it a protein structure, be it a DNA structure, be it a carbohydrate structure, or be it the assembly of lipid bilayer. So I got two interesting cartoons here. So here the water molecules are not interacting with the fat molecule. Let's just say it's that because I'm fat, right? So similarly, we have also known that there are some amino acids, because of their structure, they can very well interact with water, whereas some amino acids, because of their hydrophobic nature, cannot interact with water favorably. And this is sort of the basic process behind what is known as hydrophobic collapse, a basic step in soluble or globular protein folding, right? So similar principle works in case of membrane organization. So as I said that when there are molecules which has got part which can interact favorably with water molecule, as well as parts in that molecule which cannot interact favorably with water molecule, a unique situation arises, and that unique situation is nothing but giving rise to an organization, which is basically a setup where the part of the molecule which are capable of interacting with water are exposed to water molecule, whereas the part of the molecule that is not capable of interacting with water molecule are kept away from the aqueous environment. And in the process, what we have is a organization which is known as micelle, and the process is known as micellization. And this is our common day-to-day experience. I am sure all of you have used detergent at some point or other, and the way that detergent is able to dissolve on all the dirts from our clothes is by forming a micelles which has got an hydrophobic interior, and most of the dirt are actually hydrophobic in nature. So these micellar objects can be used to pull out the dirt from our clothes. So this is basically nothing but a you know a thermodynamic restructuring of the molecule such that the aqueous or the hydrophilic parts are interacting with the aqueous environment, whereas the hydrophobic parts are hidden away from the aqueous environment. Detergents are nothing but a long chain fatty acid molecule which has got a polar part and which has also got an extended non-polar part, which is nothing but hydrocarbon chain. Similar thing happens when we have a lipid molecule which has got both hydrophobic part as well as hydrophilic part. But unlike the detergent molecule, now this has got an additional chain, or so it is supposedly more hydrophobic, so to speak, compared to the typical detergent molecule. In fact, you might know that the typical range of CMCs, that is critical micelle concentration for a common detergent, is in the millimolar range, whereas the critical bilayer concentration, which is an analogous to critical micelle concentration, is actually in the nanomolar range. So that means the solubility of individual molecules of lipid is even less compared to its single tail counterpart, that is the detergents. So detergents actually slightly more soluble compared to lipids. So as I mentioned that the principle for bilayer formation is similar to micellization. However, in this case, when we have two-tailed lipid molecule, the organization that is formed is not a spherical micelle, is rather a bilayer arrangement. So like the micelles, here the parts that are hydrophilic or polar are exposed to aqueous environment, whereas the parts that are actually hydrophobic, that is hydrocarbon chains, are hidden away from the aqueous environment. And because of the shape of this molecule, which is roughly cylindrical, they cannot be packed into spherical micelles, but they can be very efficiently packed in a bilayer. But when the lipids are actually arranged in bilayer fashion, although the hydrophobic parts are hidden, but there is an edge problem. Edge problem in the sense that the edges are still exposed to hydrophilic environment, right? Because these sides are still the hydrocarbon chain, which has no way to interact with water molecule. So that's why a bare bilayer cannot be stable. So as soon as they form a bilayer, it wraps around itself to form a closed vesicle. So when we throw in detergents in water, we form micelle. But when we throw in some lipids, which we can very easily isolate, for example, eggs. Egg is one of those objects in which we can very easily isolate the lipid and protein. So once you isolate the protein, which is the watery stuff, all that is left is lipid, and we can very easily isolate all the lipids from egg. And if you throw in all the lipids from the egg, they will form a bilayer, and instantly the bilayer will be wrapped itself in a form of a vesicle. So we will not be able to see the bilayer structure as such in water, but what we'll be seeing is vesicles, which are sometimes known as liposome. And we will have an opportunity to talk more about different kinds of liposome later on. So as far as the membrane structure is concerned, this is nothing but a bilayer organization, which is a resultant of the clannish and xenophobic nature of water molecules, manifested in the form of hydrophobic effect, right? And the basic driving force is that the lipids form bilayer to minimize their contact with water, okay? Now, as I already told, that if we throw in some lipids in water, it actually forms micelle. And in fact, these micelles can be characterized by various techniques. For example, small angle X-ray scattering, NMR, and even slightly bigger micelles can also be seen under electron microscopes. Sometimes they can go all the way up to 100 nanometer. But when we throw in some lipids, which is basically a two-tailed counterpart of the detergent molecules, they form bilayer. And as I said, that the bilayers are not stable as such, they will wrap around themselves to form a closed vesicle. Now, this is from a thermodynamic point of view, we know that the lipids actually form bilayer. But how was that actually discovered, or rather, how was that proven? And this is one of the very crucial experiment that led to the concept of what is known today as fluid mosaic model. And that basic experiment was carried out by Grotter and Grendel. And they did something very they actually did a very intelligent experiment. So they chose a cell which has got only plasma membrane, and for that purpose they chose RBC or erythrocyte, because erythrocyte doesn't have any other membrane bound organelle. So they could very easily isolate all the lipids from erythrocyte and estimated the area of all the lipids. How? I'm coming to that shortly. And from that measurement, they argued that it has to be a bilayer, because if all the lipids from the erythrocyte is isolated and if it is only a monolayer, then the area of the lipids that they got from their experiment was actually half of the entire area. So based on these experiments, they concluded that the erythrocyte membrane, which is a representative of any cell, is actually a bilayer. So that the experiment that they actually did was on a Langmuir trough, and this Langmuir trough is an very simple device which is sort of invented by a lady called Agnes Pokel in her kitchen. How amazing that! And then Langmuir is actually modified that the primitive Langmuir trough into a more sophisticated instrument, which we now know as LV trough and Langmuir trough, and I think many of you have seen it. So in that instrument, what we can do is we can actually lay a monolayer of lipids on air-water interface. So because of the hydrophobic nature, all the tails will project out like this, shown in this cartoon, and all the hydrophilic parts will be facing the water. So, and by carefully adjusting the barrier, we can actually estimate the total area of the isolated lipid from any given species. So in Grotter and Grendel's experiment, this is exactly they did. They isolated all the lipids and they estimated the area. They knew the size of the erythrocyte, so they can calculate the surface area, and what they found is the area that is covered by all the lipids is actually double the surface area of the erythrocyte. So that led them to conclude that the lipids are actually forming a bilayer. They, however, had a blunder in their experiment, which got later corrected, but that's that mistake didn't influence the conclusion. So they are still credited for the discovery that the membrane or plasma membrane is actually a lipid bilayer. Okay, so this was the first sort of milestone in forming what is now called as a fluid mosaic fluid mosaic membrane. Okay, so here I'm going to take you through a chronicle which is only up to 2003, because this is a slightly old review written by one of the pioneer in the membrane field, membrane biology field or membrane biophysics field, Michael Edidin from the Johns Hopkins University, and he chronicled all the discovery that led to the proposition of the fluid mosaic model. Now, mind it, it's only after 2003 there has been a lot of discovery or invention after 2003, which is still happening today, and our vision of or our understanding of membrane or membrane bound organelles are constantly evolving based on these new discoveries. I will come to that later, but let us look at some of the early discoveries that happened, started from 1880s, where Lord Rayleigh and Agnes Pokel, just I spoke about her in earlier slides, they were observing the spreading of water on spreading of lipid on water surface, something that we all have seen, all have experienced. So, and that led to the characterization of how hydrophobic molecules can be spread on water surface. 1899, Overton described that a lipid-like barrier exists between cytoplasm and the outside world. In fact, Overton is the one who coined the term lipidus, and from which the lipid word has been coined. And these Overton, along with Mayer, they actually formed a sort of a rule which is known as Mayer-Overton rule, which says that the molecules which has the ability to work as anesthetic has the same ability to partition into bilayers. So that means there is already, by 1899, people are starting to understand that there is some connection to, the molecule's ability to partition into bilayer and their ability to work as a membrane active component like anesthetics. Then by 1917, Erwin Langmuir formed his or devised his Langmuir trough based on the ideas generated by Agnes Pokel, and then he is the first who could actually derive some quantitative numbers like the area of lipid, total area of isolated lipids, so on and so forth, and this laid the path for Groton and Grendel, who used the Langmuir trough to found out the area of the total lipid of erythrocyte, which led them to conclude that the lipid membrane is actually a bilayer. Then by 1935, Danieli and Davison described a model of membrane, which got refined by Robertson, and he coined the term unit membrane, and he postulated that all the membranes, all the lipid membranes in living organism probably have the same kind of structure, and he called it as a unit membrane. We'll see that structure in subsequent slides. By 1969, by this time, many of the technologies had already been developed, so people started measuring some of the dynamic features of lipids or membrane. And one of the key experiments that led to the conceptualization of fluid mosaic model is actually carried out by Edidin along with Fry, and they showed that the lipids and proteins can actually are actually mobile on the membrane surface. This is a landmark paper. I would urge you to read that. All the papers will be found in this reference given here. And then around 1972, Singer and Nicholson put up a model which is now known as Singer-Nicholson fluid mosaic model. Actually, in this, if you see the original paper which was published in Science, there is no original data in that paper. And it is not a review paper either. So what Singer and Nicholson did is, based on all the available data and their way of conceptualizing the whole thing, they actually put up a model which were able to capture all the existing data and explain them in a manner so that all the scientific community could accept it. And that gave rise to what is known as the Singer-Nicholson model. Remember, this is 1972, and the model has to be based on the data that is already available by then. So they could not have imagined something that was not known by then, right? So that's why the model is ever evolving, because the technology that led us to those discoveries are also evolving. So the technologies that led us to believe that the membrane is homogeneous is also evolved, right? So the concept that membrane is homogeneous, that is put forward in Singer-Nicholson model, has to evolve also, right? So why I'm saying this, because you will be seeing in literature that how successful was the Singer-Nicholson model in capturing all the membrane phenomena. And there are certain occasions where the fluid mosaic model was not adequate to explain. For example, the rigid domains in some of the cells, and non-homogeneous mixing, and last but not the least, the raft structure or the raft concept, which cannot be explained by Singer-Nicholson model, because the concept of inhomogeneous mixing was not there by 1972. That's why they could not capture, so that's why their model could not capture that. We'll come back to that later. After 1972, there has been a considerable development, and I'm not going to going through all of them. You will have the slides, so I can go through later. And as you'll be seeing that all the subsequent developments were basically sort of supplementing this basic Singer-Nicholson model. Okay. Are there any questions so far? If not, I'll just go ahead. All right. So as I told you that the Singer-Nicholson model was a continuous evolution of model. So the first model that for lipid bilayer that is put forward was by Debson and Danieli around 1935, and the idea that the membrane is made of lipid and proteins was known. However, how exactly they were organized was not known. Because see, by this time electron microscopy has also been developed, so people were capable enough to identify a boundary, but that boundary or the molecular organization of that boundary was not known. So this is the version of Debson and Danieli around 1935, and they thought that the proteins were organized completely exterior to the lipid. Then the next model that was put forward was by Robertson, and he sort of refined this model and said that the exterior protein is sort of integrated with the lipid, and there is no gap between the monolayers of the lipids. And these two models were there for a while till Singer and Nicholson proposed their fluid mosaic model, and which is nothing but a sea of lipids which is fully dynamic, and the proteins are embedded either in a peripheral manner or in transmembrane manner. So basically the lipids are forming the fluid, two-dimensional fluid in which the proteins are embedded in the form of mosaic, and that's why it is known as fluid mosaic model. And the fluidity comes from the part that the lipids are dynamic over a wide range of time scale. In fact, the key experiment that led to the concept of the fluidity of plasma membrane was carried out by Michael Edidin along with Fry, as I told you. Fry and Edidin is a famous paper in KCS, so I urge you to read that. All right. So now let's have a more sort of a clear or more detailed idea about the mosaic model, and this is more or less accepted model of all the membranes, particularly plasma membrane. Even the double membrane that we come across, for example, in case of some bacteria and nucleus and mitochondria, individual membranes are still oriented in a bilayer fashion, like the shown in a fluid mosaic model. So the fluid mosaic model is nothing but a two-dimensional anisotropic fluid of lipids, It's two-dimensional because the thickness of the bilayer is barely 5 nanometer. So for all practical purposes, we'll be talking about a two-dimensional membrane, right? Because if you can think of the size of a cell, which is all the way from 10 micron to 50 micron, compared to that, 5 nanometer is negligible, right? That's why it is essentially called a two-dimensional fluid because the lipids are constantly moving within the plane of the membrane, in which the the proteins are embedded, as I told you, either in peripheral fashion or in integral membrane fashion. In fact, Singer and Nicholson are the one who, for the first time, formally distinguished between integral membrane protein and peripheral membrane protein. So the peripheral membrane proteins can be very easily dissociated from membrane by changing the salt concentration, whereas the integral membrane proteins are very difficult to remove from membrane because you have to have a detergent solubilization, so to strip off all the lipids around. And in fact, Singer and Nicholson are credited for making that clear in their model. All right. So as I told you that Singer and Nicholson model is not a experimental model. It's a proposition based on the existing data, right? And it predicted things which could be, some of them could be experimentally verified, and some of them could not be experimentally verified because the model couldn't capture some of the features of membrane physiology. So one of the essential features of the fluid mosaic model is the lateral and rotational freedom of lipid molecules, which basically makes the membrane protein functional, because if the proteins are static, they are not functional, so that means we are dead. So let me give you an analogy. If we are all taken to an absolute zero where all the thermal motions are ceased, we will die, right? That's why our metabolism slowed down at lower temperature. So in order to be physiologically active, the parts of the membrane has to be dynamic, and that dynamics is supported by the dynamics of the individual lipid molecules. I'll come to that in more detailed fashion. And I already mentioned that the membranes, the way Singer-Nicholson model conceived it, is a two-dimensional oriented solution of integral proteins in the viscous phospholipid bilayer. Now, the viscosity of phospholipids, I'll come to that subsequently, but let me just tell you. It's often said that the viscosity of the bilayer, particularly in the interior of the bilayer, is more like olive oil. So if you know the viscosity of olive oil, you can have some idea about what would be the viscosity. And fluidity is nothing but the inverse of viscosity, which will be similar to what is found in olive oil. So that means you can imagine that there will be a drastic change or drastic reduction in the dynamics of lipid and protein molecules compared to the aqueous solution, which has tremendous implications in signaling. And when we change the temperature of the membrane, the domain structure changes as well as the dynamics changes, and that's why the signaling also changes. So everything is related to each other, and the key factor here is the dynamic nature of the lipid molecules. So the cartoon that I have taken for depicting fluid mosaic model is not the exact cartoon that has been proposed in the paper. You can see that for yourself. There are some components added to it. For example, cytoskeleton. In fact, in original fluid mosaic model, there was no concept of cytoskeleton. The fact that the cytoskeleton will have tremendous implication on the membrane organization was not captured in fluid mosaic model, and that is one of the drawback. And this is, I wouldn't say drawback, I would say that the enough data by 1972 was not available, and so Singer and Nicholson could not capture that feature in their model. But subsequently, as we learned more about how cytoskeleton, for example, actin and spectrin, modulate membrane organization, we have taken care of that into the subsequent models. And in fact, I'll show you the contemporary model, which is based on a hierarchical domain structure of lipid organization, is heavily influenced by the cytoskeleton. So the mobility of the lipids and proteins are also dependent by the cytoskeleton. In fact, cytoskeleton restricts the mobility of lipids and protein, and that is important for signaling in physiology. So there are some salient features of biological membranes that directly come out from the fluid mosaic model. Is that I already said it's a two-dimensional, non-covalent, cooperative, supramolecular anisotropic assembly. Okay, I have told too many phrases in one sentence. Let me take you through that. Two-dimensional is already I explained. Non-covalent means, see, all the other forms of biological macromolecules, for example, nucleic acid and proteins, the monomers are actually held by covalent bond, right? In case of lipids, in case of membranes, lipids are actually held by non-covalent interaction. So, as I told earlier, that it is basically the clandestination of water molecules which drives all the lipid molecules together. Once they are brought together, van der Waals force of attraction plays a role. But remember that van der Waals force of attraction plays a secondary role in the stability of lipid bilayer. The predominant factor is hydrophobic effect, which is a non-covalent interaction. Then it is cooperative, because one part of the membrane, if we introduce a change in one part of the membrane, that is felt in the other part because of the dynamic nature of the water molecule, the lipid molecules, because things move within the membrane or within the plane of the membrane because of simple diffusion, right? So that's why it is called a cooperative molecular assembly. Anisotropic, because the properties of the assembly is actually dependent on the direction. Even though I said it is a two-dimensional, if we go along the direction of what is called a biennial normal, the composition constantly changes. I will come back to that in the next slide. Hold on to that till then. The second point is biological membranes are formed by self-assembly, as I told you, the non-covalent force and because of the clandestination of water molecule. The third point is, since they are not held by any strong forces such as a covalent bond, they are often termed as soft matters. Now, what is the advantage of characterizing this as a soft matter? The physics which drives the interaction in soft matter can now be directly applied into membrane biology and membrane research. So that means all the the thermodynamic features that governs soft matter can be applied in membrane biology in order to have some predictive model. So those who work on the physics of lipid bilayer, they would like to call lipid bilayer as soft matters, right? Other forms of soft matters are nothing but polymers and liquid crystals. So, in fact, some of the terminologies are used in membrane are actually borrowed from the liquid crystal field. All right. So, although I'll, subsequent classes I will talk about the contemporary model of plasma membrane, but just to give you a glimpse of what are the changes that are going to be introduced in fluid mosaic model are given here. And this is also slightly dated because this was published in 2012 by another pioneer in the field called Akira Kusumi, and he along with others discovered some of the features of biological membranes which were not captured by the fluid mosaic model. So just to give you a glimpse of that, as I said earlier, that one of the basic assumption in the fluid mosaic model was that the lipids are homogeneously mixed in the form of in the plane of the bilayer, and they are free to diffuse along the plane of the bilayer. Now these two assumptions were not held true based on the subsequent discoveries. So what Kusumi and others devised a technique called single particle tracking, where they could actually monitor the diffusion of a single molecule, be it a lipid or a protein, in a plane of bilayer over a wide range of spatiotemporal scales. Right? See, this ability was obviously not there in 1972, right? But this ability was there in 2012. That's why Kusumi and others could do that experiment. So what those experiments revealed is that the lipids are not homogeneously mixed in the plane of the bilayer. They exist in the form of domains, and in fact there are three kinds of domains that the Kusumi's experiment could identify. First is a membrane component generated by partitioning of the entire plasma membrane by the membrane associated actin-based cytoskeleton in the form of a fence. So, you can think of actin or spectrin as a support to a ceiling, like, when you might have seen when we make a roof, we first make the the cage with the iron cage, and on top of that we lay the concrete. So this is similar to that. So the actins are actually forming the framework on which the concrete of lipid is laid on. So now, because of this interaction with the actin and the lipids, the mobility of the lipids are restricted, and so they are domainized. So this is one form of domain, and it is called the actin-based domain. The second is more fundamental, and that is or more thermodynamic, and again that a discovery that was subsequently been realized, that the lipids, by the nature of their structure, are not necessarily going to mix homogeneously at all the concentration. That means you take two different kinds of lipid, and give you specific example. Let's say DPPC and cholesterol. They are not going to mix homogeneously at all the concentrations. So as you keep increasing the concentration of one component, in this case cholesterol, what will happen after a threshold concentration of cholesterol, DPPC or DOPC will separate out from cholesterol rich domain. And some of these domains have been identified again by another pioneer in this field called so there is actually two scientists who could be credited for that. In fact, they work together. So one is called Kai Simmons, who is at Dresden, and then another one is called Ari Helenius. In fact, they were not looking at membrane organization and dynamics per se. You would be amazed to know that they were actually studying virus entry to the host cell. And then they found that the virus is actually budding out from only a particular direction from a cell. And that led to, the argument that the lipid composition of the cell membrane may not be homogeneous. And from that, you could, isolate the front side from and the basolateral side of the cells from which the viruses were budding out. So based on that, they put forward the idea of lipid rafts, which is basically nothing but domains or of lipids composed of glycosphingolipid, cholesterol, and GPI anchored proteins, and they are not homogeneously mixed in the plane of the bilayer. So these kind of domains are often known as rafts, because like rafts are floating in the sea or water bodies. These domains are also floating in the two-dimensional fluid of lipids. That's why they are called raft. And the third thing is the dynamic protein complex domain composed of dimers and oligomers or integral membrane proteins. So the third kind of domain is basically coming up because some proteins are actually oligomerized dimerize or oligomerize in the plane of the membrane, and when they form a cluster, they bring some lipids along with them, so those lipids are also clustered in the plane of the membrane. So now we have three basic mechanisms by which the movement of the lipids and proteins in the membrane are restricted. One is the interaction of the actin, and it is sometimes known as picket fence model. The second is the basic nature of the lipid molecules and so-called raft domains. And the third is the dynamic protein complex formation. And in fact, a lot of the signaling is actually happening because of this dynamic association of the proteins and the consequent segregation of the lipid molecules. Based on these three factors, there are now domains of different spatiotemporal scales. And this is a hotly debated topics, and the existence of these kind of domains obviously depends on the kind of techniques that you are going to use. So if you are going to use a technique which looks at membrane from more of a equilibrium point of view, you may not be able to capture all these domain perspectives. On the other hand, if you are looking at the membrane from a very instantaneous point of view or very fast time scale, like in case of Kusumi's experiment in single particle tracking, you will be seeing the dynamic nature of the domain organization of the lipid membrane. Now, these three are very significant improvement in our understanding of plasma membrane from what has been put forward in the Singer-Nicholson model. And as I told you already that this is 2012 now, and we are in 2021, so already almost 9-10 years. So there has been again another series of tremendous development in the form of paradigm shift with the discovery of super resolution spectroscopy. Now, taking that into account, we have even more sophisticated understanding of the plasma membrane of cells, and so don't take that this is the final model. It's ever evolving depending on the technological development that goes around. All right, I'll now switch gear a little bit and talk about structure of lipid membrane from a more quantitative perspective. Now, what do we actually mean by structure of lipid membrane or lipid bilayer per se? So when we say structure of a protein, we invariably imply that there is a crystal structure, and nowadays there could be an enema structure. So that means an unique organization or unique spatiotemporal orientation of a molecule. And we can have a static structure of that organization, a molecular organization, let's say either DNA or protein. That is not going to be true in case of lipid membrane. See, there are two reasons for that. When we say lipid bilayer, we are not talking about one particular protein molecule. It's a collection of molecules. Second, because the lipid molecules are dynamic over a wide range of spatiotemporal scale, they cannot be crystallized in the same fashion as proteins or DNA can be crystallized, right? However, nowadays you might have seen that a lot of membrane protein structures have come out. In that, we find the lipids stuck in the protein structure, but that's a different issue. We will get to that later. But when we talk about structure of lipid membrane, what do we actually mean, or what do we actually refer to that? So what we can do is to get a time average distribution of different components of a lipid molecule when they are arranged in a bilayer fashion from an experiment known as powder X-ray diffraction. So what we do in that experiment is we take lipids in aqueous dispersion. So when we take lipids in aqueous solution, invariably they will form what is known as multilamellar vesicles. They don't form unilamellar vesicles. There is a reason for that. Let's get into that later. So you don't have to do anything. You just throw in some lipid in water and they will form a milky appearance, and which is nothing but a multilamellar vesicle. You take this multilamellar vesicle and shine X-ray of appropriate wavelength. What we end up with is the diffraction pattern, which is nothing but a concentric spheres like this, and these are called X-ray diffraction or powder X-ray diffraction pattern, because the orientation, there is no particular orientation of these multilamellar vesicle. You get this concentric spheres. Remember, if you have seen the famous X-ray diffraction pattern of DNA, you have seen bands only at specific direction, right? Because the DNA molecule is oriented in a particular fashion. Here the lipids have random orientation, so that's why you get these concentric spheres. From these spheres you can get the intensity of this what I called a Bragg spacing, and from that we can do what is called a Fourier transformation, which will give us the electron density profile of different components of a lipid molecule. It's very similar to what we otherwise do in protein structure determination, but in a slightly different way. So we get the powder X-ray diffraction pattern, we get the lamellar repeat distance, and the Fourier transformation of that is basically the electron density profile. So the electron density profile is nothing but a time averaged distribution of different components of lipid molecule. In nutshell, that is what is meant by the structure of a lipid bilayer, and the whole thing is known as lipid crystallography, which is slightly different than how we understand macromolecular crystallography from the point of view of protein. So when we get the data and we try to put in a different fashion like this, which is nothing but a let's say a cross section of a lipid bilayer, we would end up with something like this. So these are nothing but the different components of a lipid molecule and their time averaged probability as a function of the distance from the center of the bilayer. So, and you can identify the different components. For example, the bulk water will be at the outside. After that, there will be the phosphate groups. So the phosphate groups have highest electron density because they're the most electron dense part in the lipid molecules. So that's why you see these two maxima here. And as we go along the bilayer, the electron density sort of decreases, and then it comes to a minimum, and then again it retraces back because it's actually a bilayer. So this electron density profile also reiterates the fact that the lipid structure is actually a bilayer, right? So there is another way of depicting the cross-sectional structure that I showed you is in this fashion, which is nothing but the monolayer of a bilayer. Now this is a very important figure in terms in terms of the biological and lipid organization in a membrane. What we can say from this structure is that the membrane, although I said is a two-dimensional fluid, but if we sort of have a zoomed in picture at molecular detail, we see that this is actually an anisotropic assembly, meaning as we go down along the bilayer normal, we'll see the chemical composition is actually changing. So from this structure we can see there are three distinct regions. Region A, which is nothing but the bulk aqueous phase. The aqueous phase is identified here because the water is strongly associated with the lipid head group. Then we have region B, and region B is known as interface, which is basically the functional unit of all the membrane, because all the biochemistry, or I'll be careful here, most of the biochemistry actually happens in the interface, whether it is organization of membrane protein or partitioning of membrane active molecules or drugs will all happen in the interfacial region. So this is actually a very sort of a happening space in the membrane. And then there is region C, which is more of a monotonous hydrocarbon like environment, which constitute the bulk of the membrane. Now from this structure we can immediately recognize two features of the membrane. One, there is a built-in polarity gradient, so that means as we go along from the interior of the membrane to the aqueous surface, polarity increases. In fact, we can give some numbers in terms of dielectric constant. So the region C is actually more like a hydrocarbon. So the dielectric constant will be in the range of 2 to mostly 2 to 6, something like that. Bulk aqueous region, you know, it's 78. In the interface, it is around 16 to 20, depending on what kind of lipid we're talking about, but it is not less than 16, not more than 20. So you already see there is a gradient in polarity, right? Similarly, there is also a mobility gradient. So that means things are more restricted in terms of their mobility in the interfacial region, because the interfacial region is capable of forming a hydrogen bond network, and that's why they are interacting with water molecule. Remember my initial slides where I showed that the part of the lipids which are capable of forming hydrogen bond network with water, they're also capable of forming hydrogen bond network with each other, and that's why the mobility of the molecules are restricted in the interfacial region. But as we go down, there is nothing that are holding the molecules together except the van der Waals force. So things are more mobile in that sense. More mobile in the sense they have more rotational and vibrational freedom. So there is also a mobility gradient. So there is both polarity gradient and a mobility gradient. In fact, they go in opposite direction with each other. And these two are very crucial factor for the organization of the membrane proteins. So that means the membrane protein composition should be such that they are compatible with this polarity gradient, right? So that means that all the hydrophobic amino acid has to be placed in the interior of the membrane, whereas the polar amino acids or the charged amino acid should be in the membrane interfacial region. So when there is mutations and this organization is not maintained, the functionality of those membrane proteins are severely hampered. One classic example of that is cystic fibrosis, where the cystic fibrosis protein has a lot of these mutations where you get a charged residue in the middle of the membrane or a hydrophobic residue in the interfacial region. So they play havoc in the functioning of these proteins and lead to severe genetic disorders. All right, now few words about this mobility gradient. I will come back to that later in the form of the membrane dynamics, but this mobility gradient is over a wide range of temporal scales. So this mobility gradient can be identified by fluorescence spectroscopy, can be identified by ESR spectroscopy, can also be identified by NMR spectroscopy. Now, as you might know, that between these two techniques, we already talked about 3 orders of magnitude sub time scale, meaning fluorescence is a nanosecond phenomena, ESR or EPR is a microsecond phenomena, NMR is a millisecond phenomena. So all the way from nanosecond to millisecond, this mobility gradient is maintained, and that's why we are able to capture this mobility gradient by diverse technique, right? So this is not a very transient feature of the membrane. In fact, this is a integral part of the membrane which is manifested over a wide range of temporal scale. All right. So so far I have been talking largely about how the lipids are organized in the form of a bilayer and what are other properties that actually emerge because of that particular organization. For example, this polarity and mobility gradient. In fact, these two play a huge role in membrane remodeling and curvature later on, and we'll talk more about that. By the way, these things are known as emergent properties because the individual molecules will not have this particular feature, but when they come together to form a particular molecular assembly or organization, these kind of features or properties emerge. That's why they are called emergent properties. In fact, membrane potential, curvature stress are all emergent properties. All right, so I wanted to reflect a little bit on the composition of the membrane, meaning what are the different kinds of lipids that are found across different membranes and different species. This is in fact a slide taken from a textbook. So this gives you a more, a comprehensive view of what are the different possible lipids. The first one is of course storage lipid, and in fact we have been talking about lipid droplets. So basically the fat, what is stored in lipid droplets is the fat, and the fats are nothing but triacylglycerol, the precursors of membrane lipids. And as the name suggests, the triacylglycerol, they are basically esters of glycerol and fatty acid. From that, we can think of the structure of lipid molecules. So lipids, or the membrane lipids, which are abundantly found, can be largely classified into two categories. One is called the phospholipid, another one is called the glycerol lipid. Now the phospholipid can also be classified into two categories, what is called a glycerol phospholipid and a sphingolipid. Now the difference comes in based on what kind of backbone the lipid is made of with. In glycerol phospholipid, it's a glycerol backbone. So as you know, that a polyol glycerol, and which is nothing but an ester with fatty acid, and then there is a phosphate group, and sometimes there could be an alcohol or other small molecule like phosphatidylcholine. So this constitutes the large amount of lipids that are found in nature. Then the second kind of lipid is known as sphingolipid, and the major difference between the glycerophospholipid and sphingolipid is in the backbone. Unlike a glycerophospholipid, the sphingolipids has a backbone which is known as sphingosine, and sphingosine molecule has already a long fatty acid chain associated with it, so it has got another fatty acid chain, and then a phosphate group, and of course a choline and phosphatidylserine, and depending on that there will be different kinds of sphingolipids. Similarly, there will be lipids called glycolipids, where instead of the phosphate group, a sugar moiety will be attached, and these different kinds of sugar moieties can be attached, and then they are largely called glycolipid, and a subcategory of them is known as glycosphingolipid. And in fact, glycosphingolipids are very crucial, as you might know that some of the blood group antigens are nothing but a particular sequence of glycosphingolipids. And these are also heavily involved in signaling. So phospholipids are largely known as structural lipids, to some extent sphingolipid. Glycerophingolipids or glycolipids are mostly known as signaling lipids. There is another kind of lipid which I do not have in this categorization, and that is called phosphatidylinositol phosphate. They can be thought of as largely as glycosphingolipid, but they have, so this is again a separate class of lipid by itself, and they are their abundance is relatively low compared to typical phospholipid or sphingolipid, so they are mostly considered as signaling lipids, and these are called phosphatidylinositol phosphate or PIPs. And you might have come across PIPs in different contexts because they are heavily involved in signaling. There are different kinds of lipids which are not found in bacteria or higher eukaryotes. They are exclusively found in Archaebacteria, and they are called Archaebacterial lipids or bolalipids. One of the interesting features of these lipids are they are actually they don't form typical bilayer like the sphingolipids or phospholipids do, because they don't have any middle plane like the sphingolipids or phospholipids do. So they are both ends are actually connected like shown in this schematic. So this is another important distinction of archaebacterial lipids from the bacterial lipids or mammalian lipid is that the linkage between the glycerol and the fatty acid is an ether linkage. In case of phospholipids and sphingolipid, it's an ester linkage, right? As I said, that lipids are nothing but ester between glycerol and fatty acid, right? That is not true for archaebacterial lipids or bulla lipids. And in fact, there are some structural differences in archaebacterial membrane compared to other bacterial membrane or mammalian membrane. And some of these structural differences are actually manifest in the proteins that are found in archaebacteria. There are another class of lipids, and these are mostly called neutral lipids, and these are known as sterols. Now sterols, as you might have known, has a very different structure compared to either glycolipid or sphingolipids, but they still have a polar part and a non-polar part. Although sterols by themselves cannot form a bilayer, however, they can nicely be integrated in the bilayer and modulate the properties of lipid bilayer. So sterols play a major role in the organization and dynamics of lipid bilayer membranes in different organizations, right? Now that I just wanted to mention, we already know about genomics and proteomics, and in a similar manner, the branch of the omics, which deals with lipids and basically identification of different kinds of lipids in different species as well as in different organelles in normal and pathophysiological condition, is known as lipidomics, which is basically lipidomics is identification of new lipids by various kinds of mass spectroscopy and their quantification and association with any kinds of pathophysiology. So it's an emerging field, and it's probably right time to be involved in lipidomics research. All right, just to give you a basic idea about the lipid structures, as I already mentioned, that the glycerol phospholipid is based on the ester linkage between the glycerol and the fatty acid. In case of sphingolipid, this is a sphingosine moiety which already has a fatty acid chain associated with it. So it's just another fatty acid attached to it with an amide linkage, of course. So these are two major structural differences between glycerol phospholipid and sphingolipid. Sterols, on the other hand, are all a different class by themselves, but as I said, that it still has the features of a typical amphiphilic molecule, for example in a head group which is characterized by this OH group, and a large nonpolar group which is made by the fused steroid ring, right? So they, as I said earlier, they themselves cannot form a bilayer. However, they can be integrated into bilayer, and then they modulate the various properties of the lipid bilayer, including the fluidity of the lipid bilayer membrane, which is an important factor for the functioning of lipid membranes, right? Depending on, there is okay, I'll just wanted to mention briefly, there is one particular class of lipid which is nothing but as if two phospholipid molecules stitched together with an additional glycerol molecule, and this is known as cardiolipid, and this is exclusively found in mitochondria, and this is also known as a mitochondrial lipid. I should mention that although I have been saying that the linkage between the phosphate group, the glycerol backbone and the fatty acyl chain, or the sphingosine moiety and the fatty acyl chain, either ester or amide, there is a class of lipid called plasmalogin in which one of the linkage is ether and the other linkage is ester. So plasmalogen in that sense is kind of a hybrid lipid, again, which is not exactly a structural lipid and found in under specific situations. Okay, so just keep that in mind, that not all the lipids are exclusively formed by distal linkage or amide linkage. Sometimes there could be a combination of the two, for example ether and ester in plasmalogen. And this is the structure of representative archaebacterial lipids. And you might notice that the fatty acyl chain also look different. They are mostly saturated, and one of these side chains, in fact, the amide, the lipid that we typically see in bacteria or higher eukaryotes do not have these kind of fatty acid composition, and also do not have this ether linkages as such. So, in case of archaebacterial lipids, you don't have to form a bilayer. The lipids can be simply arranged in a monolayer fashion. Okay, and because of these particular arrangement, there are some structural differences, and I think in the next class I will probably briefly touch upon that, and I had an opportunity to work with some of these lipids, so I'll share my experience in that front. All right, one of the things that we have to appreciate is that although there is a great repertoire of membrane lipids, and in fact although the different kinds of lipids can be summarized in one slide, you can imagine the kind of variety that you can generate from these moieties by different combination. So there could be n number of different ways that you have the fatty acyl chain composition can change. You can have different ways of arranging the membrane head groups. So in other words, there will be different kinds of lipids that are found in different organelles as well as different organisms. So for example, in this slide, in this slide it is actually shows that between two species, that is yeast and typical mammalian cell, how each organelle has a different lipid composition. So, and this slide also shows that most of the lipids are actually synthesized in the inner organelles. For example, either endoplasmic reticulum or Golgi, or sometimes some of the conversions between the PIP1s to PIP2 also happens in late endosome. So the lipids are actually synthesized in some of the inner organelles, for example ER or Golgi, and from that they are actually transported into the plasma membrane. So plasma membrane is not a site for lipid synthesis. There may be small modifications, for example when we have a phospholipids treatment and so on and so forth, but this is not the site for lipid synthesis. In this series of lectures, we may not have the ability to talk in detail about the lipid synthesis, but just remember that the lipids are synthesized in ER or Golgi and then transported into the plasma membrane with the help of various lipid transfer proteins. Okay. Now this slide also shows that the composition of the lipid membranes are also different. For example, the ER lipid composition will be very different from the plasma membrane lipid composition compared to mitochondrial lipid composition. Similarly, late endosomes, which are dynamic vesicles, will have a different composition compared to early endosome or lysosome, right? And which can be seen from these schematics as shown in this figure. One of the key parameters is the cholesterol to phospholipid ratio, and that varies from membrane to membrane. For example, Golgi has a very low cholesterol to phospholipid ratio, 0.2. ER has a slightly higher and slightly lower 0.15, whereas plasma membrane sometimes have 50-50, 50% cholesterol and 50% phospholipids. So cholesterol to phospholipid is almost like 1. So this is a drastic difference, and that difference shows up in the membrane thickness. So one of the ways that the lipid composition modulate membrane structure is by changing the membrane thickness. You can imagine that the bulk of the membrane is actually the hydrophobic chain, right? So depending on the acyl chain composition, how long and how short it is, the bilayer thickness will be different. And the bilayer thickness actually plays a profound role in the organization of membrane proteins, which I will elaborate in the next class. But the bilayer thickness is one of the crucial structural parameter of any plasma membrane, and which is strictly modulated by the lipid composition and the cholesterol composition in it. Cholesterol has the ability to organize the lipids such that their vertical length is maximum, so that you have the maximum bilayer thickness when there is a significant amount of cholesterol, and that thickness of the bilayer will be reduced significantly if you have low amount of cholesterol. In fact, from Golgi to ER2 plasma membrane, the bilayer thickness is gradually decreases. So that's why the typical proteins that are found in ER has a shorter transmembrane domain compared to the proteins that are actually found in the plasma membrane. This plays a very important role in sorting of proteins. I think I'll elaborate on that in the next class. I just wanted to briefly touch upon one structural aspect of the lipid membrane, and that will be my last slide today, and that is the phase of the lipid. So mostly the lipids in the bilayer fashion are organized in what is called an L-alpha phase, or sometimes it is known as fluid phase. But there is another phase, and which is known as L-beta, and as I told you earlier that the lipid membranes share similarity with soft matter. So the L-alpha, L-beta are the terminologies that are used in liquid crystal field. So since the liquid crystal also shows this kind of phase behavior, these terms are borrowed from that literature. Okay, so typically the lipids are oriented in a bilayer fashion in a fluid state, and which is known as L-alpha phase. Sometimes it is also known as a liquid crystalline phase. And in that structure, or in that phase, the lipids are kind of relaxed and loosely packed, and that's why they are more mobile. On the other hand, if we slightly lower the temperature, the lipids will found themselves in a different phase, and that is known as a gel phase or L-beta phase. In other words, it's also known as an ordered phase. So L-alpha phase is also known as a disordered phase. L-beta phase is also known as a ordered phase. So this transition between fluid to gel is also known as an ordered-disordered transition. And this is a thermodynamic phase transition. That's why it can be very easily captured by thermodynamic methods, for example, DSC or differential calorimetry, differential scanning calorimetry. There are other methods as well to detect this phase transition in the bilayer. In subsequent classes, I'll discuss that briefly. One of the main feature of this so-called phase transition in the bilayer, that is after a specific temperature, above a specific temperature, all the lipids will be more relaxed and they will be more dynamic in fluid phase. On the other hand, below this particular temperature, which we call as phase transition temperature for a particular lipid composition or for a particular species, In that, the lipids are very closely packed or very tightly packed, and their mobility is kindly restricted. So depending on the lipid composition, a given membrane will either be in a fluid phase or in a solid or ordered or gel phase. Depending on whether it is in gel phase or in a fluid phase, the mobility or the dynamic features of the membranes will be different. And this is a very crucial factor because, as I told you, that in order for the membrane proteins to be perfectly functional, they have to be in a fluid kind of environment. If you make it in a very rigid, the proteins may not be able to function. So that's why the membrane lipids will be constantly under metabolic control such that its fluidity is constantly maintained depending on the temperature of its environment. And it is more relevant for those organisms which cannot maintain their body temperatures, right? And this feature is known as homeoviscous adaptation. So the organisms which are referred to as poikilothermic organisms which cannot control their body temperature, they must adjust their lipid composition such that their membrane will be perfectly functional so that they can withstand the environmental stress. And this is known as homeoviscous adaptation, which basically tells that the cells has the ability to control its metabolism such that the membrane fluidity is maintained at optimal level so that the organisms don't suffer any stress and they can be they can survive under adverse conditions. So this is it's not that we understand it completely, but the for some organism this has been studied very well. For example, yeast, we can grow yeast at different temperature, and depending on the temperature its fatty acid compositions will be different, right? I forgot to mention one part, that the phase transition temperature actually dependent on the composition of the fatty acyl chain. It's dependent on the level of saturation as well as dependent on the acyl chain length. As a thumb rule, you can remember that if we have more unsaturated lipids in the bilayer, we have a lower phase transition temperature. So more unsaturation, more fluidity, less unsaturation, less fluidity, and higher phase transition temperature. So if your membrane is exclusively made of saturated lipids, it will have a very high phase transition temperature. On the other hand, if it has a significant amount of unsaturated lipid, meaning double bonds in the acyl chain, it will have a very low phase transition temperature. For example, POPC or DOPC. DOPC will always be fluid at room temperature. DOPC's phase transition temperature is actually -20. On the other hand, if you take DPPC, which is dipalmitoyl phosphatidylcholine, which is made of all saturated lipid, it has a phase transition temperature of 25 degrees Celsius. So if you take DPPC lipid membrane at 25 degrees Celsius, below 25 degrees Celsius it will be gel, above 25 degrees Celsius it will be fluid. So and that transition temperature can be very well studied by a thermodynamic method called DSC or differential scanning calorimetry. Okay. I think I have taken more than what I supposed to, so I'll stop here today and I'll continue tomorrow. So I'll briefly sort of revise. Yeah, we will see that, definitely. We will hear that, Sudhir, in subsequent classes, of course. So I'll stop here. Are there any other questions?
Any questions from the participants?
So far, I will talk about how proteins shape membranes, and in fact, that's all about membrane remodeling.
Maybe for the audience, like we discussed about the phase transition.
Yes.
Yeah, so like say in a normal physiological conditions, like say the fuse behavior is very important for like say providing kind of a signaling platform as well.
Yes,
So unlike the temperature contribution and like say definitely acyl chain is like say the predominant, which is going to like say recruitment of the different lipids like a PIP2 and those means how do you interpret like say means what can be the driving force under physiological condition to uh, uh, adapt the membrane, bind the membranes to segregate into the liquid ordered and a disordered state. And that
yes.
means yeah, yeah.
I'm glad that you brought out that question. See, all the ideas of the lipid phase transition, so they are very good when you have pure lipids. But think
Yes,
of a
yes.
physiological condition where we have different kinds of lipids, um, different kinds of membranes, and they're constantly influencing each other. As a matter of fact, in typical membranes, we do not see a pure gel phase or pure fluid phase. What we see
Mhm.
sometimes is known as a liquid disordered phase
Yeah.
or liquid ordered phase. In fact, raft is nothing but a liquid ordered phase. So it's a kind of a sort of a middle ground between the complete liquid disordered phase and the solid ordered phase. That's why it is called a liquid ordered phase or LO phase. So if you go to my previous slide where I was showing you L alpha and L beta, and these are fine. You do see some L alpha, L beta in some membranes, but mostly what we will be seeing in a typical cell, typical mammalian cell, is under reasonable cholesterol concentration is LO phase, what is known as a liquid ordered phase. So
Mhm.
we you can still see a phase transition, but that phase transition will be very local. And you can modulate that phase transition by again modulating the phase transition, by modulating the temperature and lipid composition. For example, if you
Exactly.
modulate the lipid composition by either chronic way or acute way, meaning either you can, take out cholesterol by methyl beta cyclodextrin. That's the
Mhm.
acute way of changing lipid composition. Or you can treat with some inhibitors of cholesterol synthesis, that is more chronic way of doing it. In both cases, you will see that the phase of the membrane will be different, right? You may not see a very sharp phase transition as you would have seen it in model membrane, but you will still see some changes in the dynamics of the lipids as well as in the proteins which are associated with those domains.
Exactly. Yeah.
In
And then
fact,
I was,
yeah,
yeah, go ahead, go
what
please.
I think I can bring out a movie tomorrow. You can actually see how, so we may not have the phase transition as such, but what will happen is coalescence of domain. So if you reduce the temperature, what you will see that the domains are merging and they become more homogeneous. And on
Hmm.
the other hand, if you reduce the temperature, you'll see the domains are kind of segregating from each other. This
Exactly.
can be nicely shown in cells, but if you can get rid of some of the cellular influences and make blebs from cells, you can very easily
Exactly.
see that.
Yeah, yeah, yeah,
Um,
yeah, yeah, yeah.
in fact, some people are very actively studying it. Earlier, Leventhal, and in fact, Kai Simmons' group is also actively involved in that.
Uh-huh, uh-huh. And one I was very fascinated by your, like, say, the talk. I think means there's a lot of heterogeneity in the membrane lipids,
Yes,
and all through across the evolution, we see, like, say, certain lipids have been excluded. Means it all through the evolution, not means they do exist in the archaebacteria, means the different bacteria, and then like say evolution is being to the mammalian system. So means what a kind of like say means the advantage of excluding such lipids and what's the advantage first of all excluding such lipids, and then what may be the like say the synonymous lipids which are going to replace in the mammalian system, or the one thing, and does it impact the means the structural organization of such lipids, like say means the one which we have like say older, I mean because you already have, I think probably you will be discussing about it, but in general I was just curious to
yeah.
know about it.
Yes.
How through the course of evolution that is like say means we are losing
That is a very, I would say that's a great question, and I don't think I have a clear-cut answer to that, because it's very difficult to predict why in the evolution there is a different field of lipid which is only found exclusively in archaea, and there is a altogether a different bunch of lipids which are found in hydrobacteria.
Even like say many bacteria also means
Yeah,
like say, mycobacteria or those sort of things, the pathogen as well.
yes. So what I can say is that how is that difference, um, or in more straightforward way, what are the differences? And we can probably hypothesize that these differences may play a role in the functioning of that particular proteins which are found. For example, see the dynamic gradient that I showed you in one of the slide for typical phospholipids. So you have a mobility gradient and you have a polarity gradient, and they go sort of opposite
Vice
to
versa.
each other.
Yeah.
That is a resultant uh, or a sort of resultant emergent property because of the bilayer arrangement, right? Uh, and because of this bilayer arrangement, the interior of the lipid molecules are kind of free. So they are free to rotate. But now you can imagine when you have stitched them, they are also rigid like their interface.
Exactly.
So what I have been able to show that if you have a bilayered lipid, you know the typical phospholipid have a mobility and polarity gradient. The same gradient actually do not exist in archaebacterial lipid because of this stitching. So this is more like a monolayer of the lipid. So as you walking down along the bilayer membrane, bilayer normal, you don't see the dynamic gradient that you see in, or the polarity gradient that you see in the typical phospholipid.
And I think what I assume like say it's providing kind of an stability also for the
Yes,
functional
and
stability.
that's
And I think
why,
and I think like say during the course of evolution, such stability is being provided by the different proteins or by the like say
yes,
means the lateral mobility, what we talk about, like say the rafts, and also by the ectomyosin mean
yeah,
at the cell. I mean those until those stabilities are going to provide it to the membrane, at least for the functional as well as for the signaling domain.
definitely. One plausible hypothesis would be, since the archaeas are sort of evolved in more hostile environment than the typical eukaryotes, that's why they needed probably more stability in their lipid membrane. So that,
Exactly.
uh,
Yeah.
in fact, these ideas are being exploited in making vesicles with lipids that are isolated from archaea for some biotechnological applications, so that they can survive extreme conditions.
Like the proteins, like I mean, certain
Yeah,
like thermophilic proteins are there.
yeah. One
Oh,
of
that's
the
great.
challenges to isolate those lipids in the.
Yeah, that I can understand. I see one question popping up here from I think
Oh, of course,
Dr.
aging has a very profound
Uh-huh.
effect on cell composition. I'm glad that you asked. Yeah, it's obvious, right? So I can give you one example. And again, it's cholesterol. So as the cholesterol level changes as we grow or age, whatever it is, membrane properties are gradually changing. So if, for example, if you have very low amount of cholesterol, then it's not good for the membrane, because you need to maintain a certain kind of organization and uh, let's say fluidity for the membrane and certain other properties. For example, there's something called dipole potential and curvature stress. So all will be optimal if you have a particular level of cholesterol composition. And as an individual ages, the cholesterol level also changes, and that's partly because of the metabolism, partly because of the food habits, and so that now imagine all those factors which have been maintained at a different level because of a particular sterol or particular cholesterol concentration. When that concentration is no longer maintained at the same level, all those parameters will be influenced, right? And naturally they will reflect in the uh, functioning of those membrane proteins and their consequent physiology. So yes, answer to your question is, of course, aging has a profound effect on the membrane composition.
So I don't see any additional questions. So
Protein-protein interaction, I will be talking a little bit in the next class.
So yeah, you were saying something.
Yeah, so what I will do in my next class, I have a couple of more slides left regarding lipid dynamics. So I wanted to give you a flavor of dynamics and some of the techniques that are used, in order to study those dynamic features, and then move on to lipid-protein interaction.
Yeah,
and,
it's all. Yeah, whatever way you like. Yeah, you are the one of the biophysical experts. So we will just leave it to you. Yeah. So if there is no additional questions, then I would like to thank Dr. Sourav for his time and the wonderful journey to the membrane means the basics of the membrane biology. Really, like say means we as such has been ourselves has been learning a lot, and I think most of our students might be benefited with this lecture, and like the follow-up lectures going to be taken by him. So yeah, thank you, Dr. Sourav, and thanks for your time.
Sure.